control. How to control the plant. These are things we are discuss about to the today topic. These are the course introduction. The total course, how to do, what is the course objectives, and these are things we will discuss about today class. Here, the plant pathology. Plants are the primary source of various materials like food, cloth, house, building materials, drugs, etc. These plants or plant products may be reduced in the quality by the diseases in the field or drinking water and drink storage, which can be controlled by the knowledge of plant pathology. The plant pathology deals with the detailed study of the plant disease and their control methods in the field and storage. Thus, the objective of plant pathology is reducing the loss in the yield of crop to its minimum. Keeping this in mind, the present block is divided into following 5 units. That is, the first one is the introduction of plant pathology, covers history and significance of plant pathology and pathogenicity, postulates, classified of plant disease, and importance of plant pathological terms. The second one, symptoms of the plant disease, covers various symptoms produced in plants by the fungi, bacteria, and viruses. The Unit 3, control of plant disease, cover various methods of disease control like prophylactic and therapeutic and immunizations. The Unit 4, disseminations of pathogen, epidemiology, and diseases forecasting, covers how pathogens are disseminated in environmental and various factors of epidemiology and disease forecasting. 5, disease of plant covers some important fungal disease of croplands. Objective after studying this block, you will able to known as coach postulates, which help you in identify diseases. How knowledge about various symptoms produced by pathogen. Understand dissemination and epidemiology of pathogen for disease forecasting. Known various methods for control of plant disease. First one is the introduction, objectives, and first one, introduction. The term pathology derived from the Greek word pathos meaning suffer, logos meaning study. The suffering, logos meaning study. Hence, pathology is the study of suffering. Phytopathology. Phyton is plant, pathos is the suffering, logos is knowledge. Is the study of suffering plants and deals with the study of nature. Nature, development and control of plant diseases. The disease plant is the central theme to which are interlinked the pathogen, the environment, and the time period. According to modern concept, disease is an interaction among the host, pathogen, and environment. The disease plant can be diseases, which are brought about by the unfavorable environment or by parasitic agents. Sackman and Harrier, 1957, defined plant disease as physiological disorders or structural abnormality that is harmful to the plant or to any of its part or reduce the economic value. The study of the unit able to history and signification of plant pathology, post-postnatal pathogenesis test, classification of plant diseases, various terms and required for pathological studies. What is the history of the pathogen plant pathology? The advent and progress of human civilization is intimately associated with the progress of agricultural, but ever since man started depending on the plants to fulfill his nutritional needs, the enemies of plants have been causing much harm and economic losses. Records of parasitic fungi data back to Devonian period, which suggested that disease originated along with the plants. In Indian mention of the plant disease has been made in Rigveda, 1500 to 500 BC. The Artha Sahasra 321 to 186 BC. Sustra Samhita 200 to 500. Agni Purana 500 to 700 AD, etc. In Europe, Theophrastus, while describing trees, cereals, and pulses, also recorded the harmful effects of wind, weather, and isolation, and believed that diseases originated from plants to form the environment. But the invention of the microscope changes the entire concept. Mitchell in 1729 made an extensive study of fungi and their reproductive structures and experimentally provide that the fungi originated from spores. Till late 1755, demonstrated that wheat seeds dusted with burnt disease of wheat was caused by transmission of black powder, burnt and was contagious. The fundamentation of modern plant pathology as a science was established by the French scientist Prevost in 1807, who demonstrated that microorganisms are disease causing. Working with burnt of wheat, he studied the germination of spores and described the life cycle of the burnt fungus in details. In an important discovery made up of Prevost was the fungicide and fungistatic properties of certain chemical, but due to the prime belief sciences, scientist in the theory of spontaneous generation, Prevost discoveries were knowledge 40 years later. The devastating potato disease in the middle of the 4th century, which swept over the whole of Europe and Ireland, was one of the most tragic events in human history. But it was a blessing in disguise as it compelled attention of scientists on the importance of plant disease. The casual organism of the potato disease was identified by the Spirchilier and DeBerry Phytophthora infestis, but it was believed that the fungus developed from the disease rather. The role of fungi in disease development and observation of the various scientists was recorded by Kuhan in 1858 in the first book of plant pathology. A big step forward in the field of plant pathology was made by the Brifield of German scientist in 1875, who developed modern techniques of the inoculation and artificial culture of microorganisms. Parasitic fungi were now being cultured in sterile synthetic media to study their physiology nutritional requirements, sporulation, and genetics. Grape cultivation was the main industry in the France for winemaking. In 1878, the downy middle of grape wine wiped to the crop. Professor Millardet of Bordeaux discovered that a mixture of copper sulfate and lime, when sprayed on grapevine, prevented the mildew disease, and the Bordeaux mixture was invented. Thus, the method of chemical control of plant disease initiated by Prevost in 1805 was firmly established. The variability of parasitism in pathogen and the existence of the hello. అవునా అసిస్టెంట్ ప్రొఫెసర్ సేమ్. సేమ్ టాపిక్ ఇద్దరిది. ఆ, అక్కడ మీది కూడా రాసుకోవచ్చు. మన ముగ్గురు పేర్లు పెట్టినా గుర్తుందా? ప్లస్ స్టూడెంట్స్ ఇచ్చింది కూడా మన ముగ్గురు పేర్లు వస్తాయి చూడండి. ఆ, సరేనా? ఓకేనా? సరే. ఉమ్. The basilic and in the parasitism pathogen extant physiological race of rust fungus was discovered. Swedish scientist Erikson in 1894. In the first decade of the 20th century, scientists studied the importance of genetics in plant disease resistance and selection of the resistance lines was introduced to obtain resistance variety before in 1903 or 1909. At the same time, war in 1903, Salmon discovered physiological specialization in rust and powdery mildew of cereals. Black Sea in 1904 reported that dissimilar nuclei take part in the life cycle of fungi. Fusion between dissimilar nuclei within a cell was demonstrated by Burgess in 1912 to 1914, and the term heterokaryosis was given in the phenomena, that is the Black Sea heterophenomena, heterokaryosis. Hansen and Smith in 1923 established the theory of origin of physiologic races through heterokaryosis. Floor in 1955 advocated the important gene-to-gene hypothesis of disease resistance and susceptibility, which stated that disease susceptibility depends on compatibility of genes between the host and pathogen. In early developmental stages of the plant pathology, scientists concerned concentrated only on fungal diseases, and bacterial association with the disease was not taken into consideration. But Louis Pasteur and Robert Koch in 1876 proved that anthrax disease of cattle was caused by a bacterium. Later in 1878, Burley reported that the fire blight of apple and pear was caused by the bacterium, and by the year 1900, E.F. Smith firmly established bacteria as important disease causing the pathogen. The discovery of the tobacco mosaic virus, Mayer in 1886, was the birth of the study of various viruses as pathogenic agents. He demonstrated that the mosaic disease could be transmitted from diseased plant to healthy plant by infecting the sap of leaves showing mosaic symptoms. Ivana Iski and Bizarink. Both are the scientists from the identification of the viral. Ivan Iski and Bizarink are credited for starting study on the nature of viruses. In 1892, Ivan Iski demonstrated that tobacco mosaic virus, that is called the TMV virus, could pass through those filters that retained bacterial cells and suggested that viruses were smaller than the smallest bacterium. In 1998, Bizarink coined the term Contagium vivum fluidum, that is called the infection living fluid, that is called the CVF. Potassium vivum fluidum infection living fluid, and observed that new growing tissues were more easily infected than old tissue. In 1935, Stanley experimentally proved that viruses could be that viruses could be chemically treated and crystallized and still remain viable. The following year, Bowden and his co-workers found that the crystalline powder of the viruses contain nucleic acid and protein. And in 1956, Grieger and Sturm demonstrated that the nucleic acid fraction of the virus is actually the infectious agents. Needham in 1743 AD for the first time reported the disease causing nature of nematodes. But it was century later that Breckley and Scratch discovered root knot nematodes and cyst nematodes of the sugar beet. Now almost every crop is known to be attacked by one or more of nematodes, and nematology has become a major branch of plant pathology. Knockard and Rocks in 1898 discovered another group of disease initiations which was similar to viruses in size but could not be cultured. This group was known as Mycoplasma. Japanese scientists in 1967 discovered the Mycoplasma-like organism could be responsible for most alloying disease supposed to be caused by the viruses. The plant pathology in the 20th century, in the later half of the century, the plant pathology progressed in several directions. Physiology of plant disease, the study on physiological aspects of plant disease was initiated by D. Berry and pursued by Johns, Brown, Goman, Walker, and others. The important role played by the fungal enzymes and toxins was established. The second one, genetics of the host plant and the pathogen, inheritance of the resistant, and virulence is related to the genetical constituents of the pathogen. Floor in 1955 explained host-parasite relation in flax rust by the gene for gene relationship between rust reaction in the host and pathogenicity in the parasite. Third one, fungicide research. Several biochemists and organic chemists, several biochemists and organic chemists joined plant pathologists in evolving new fungicides. Sisler, Cox, Ludwig, and other workers explored the mechanism of fungicide reactions, and in 1935, hmm, Tisdale and William discovered alkyldiothiocarbamates, the most widely used fungicide. The discovery of glyotoxin by winding and immersion from Trichoderma viridor that prevented damping of citrus seeding opened the field of antibiotics. Most antibiotics are bacterial. However, gylotoxin, petulin, and griseofelvin are used against pathogenic fungi. Fourth one, environment in relation to disease development. Environment is an important factor influencing disease development and includes climate, epidemic and biotic factors. Next coming fifth one, nature of disease resistant. Disease resistance is associated with phenols processing antibiotic properties. Muller proposed the post-infection formation by phytolauxin, chemicals which produced in non-host towards of the pathogenic fungus and provide infection by other fungus. The sixth one, biochemistry and physiological of the disease state of the host. The various metabolic process of the infected plant have been investigated to determine the sequence and extent of the pathogenesis. In the disease state, the tissues of the host disintegrates, water become deficient, growth and reproduction are adversely affected. Host starvers and respiration rate is already, hence the entire physiological and biochemistry of a diseased plant change. Tissue culture, the technique of the tissue culture enabled plant pathologists to grow organs, tissues-free cells and to study the transformation of a normal cell into a tumor cell as a result of infection. The first artificial culture of a forest fungus and micro-free culture of viruses and nematodes were done by this method. Eighth one, ecological studies of the plant pathogen Waxoman in 1932 emphasized the important role of fungi in soil economy and decomposition of the organic matter. Rinking and Mans in 1933 coined the term soil inhabitants, soil inhabitants to fungal forms occurring in all type of soil and soil inverters to flow from restricted to few locality few localities only. The interesting field of rhizopus studied was initiated by Hiltner 1904 and Lockheed 1940 demonstrated that rhizosphere flora affected the resistance and susceptibility of plants to root infection. In 20th century, plant pathology and discovered plant pathology had developed into a multi-branched sciences, embracing all segments of the biology and the entire human race is obliged to the plant pathology plant pathologist for their important contribution in ensuring disease-free nutritional food supply. What is the high-rich Anton de Berry? German surgeon, botanist, microbiologist, and mycologist is considered father of plant pathology as well as founder of the modern modern mycology. Some important plants of India are E.J. Butler, B.B. Mundakar, K.C. Mehta, T.C. Sadashivam, R.C. Tandon, S.P. Raya Chuduri, G. Raya Swami, S.N. Das Gupta, etc. Sri Edwin John Butler, Irish microbiologist and plant pathologist, is considered father of modern plant pathology in India. He studies in India about 20 years was made up of made census, made scientific studies of the most of the diseases known in India at that time. Butler left the India in 1931, 1920. V.B. Mudkar started work on cotton wilt and started Indian Phytophysiological Society. Sri S.K.C. Mehta and S. Pradhan worked on epidemiology of Central Rust in India. The next one, T.S. Sadasivan established the School of Mycology and Plant Pathology at Madras University and laid to the foundation of research on soil become pathogenic India. What is the significance of the plant pathology? The plant diseases are important as a causing damage to plants and plants products. The disease significance, importance of the diseased plants, diseased or damaged plants and plants products. The extent of the harm caused varies with the plant, the pathogen locality, environmental and environment, etc. The plant pathology is concerned with all the aspects of features of the plant disease, causing by the different microorganism, fungi, for example, fungi, bacteria, viruses, nematodes, and parasitic higher plants. Higher plants, etc. The study of the plant pathology of special significance as it makes us knowledge not only about about the various kinds of disease infecting plants, but also explain about the measures of control and prevention to be taken against those diseases. Plant pathology is not isolated subject, but it is intimately associated with botany, mycology, virology, bacteriology, plant anatomy, plant physiology, plant genetics, biochemistry, soil science, and other branches of sciences. Pathogenesis, the potential capacity of infection of any pathogen is called it is pathogenicity. The index of the primarily termed as the infected index of the quantity nature of the pathogenic microorganism is called is called the degree of pathogenic or the degree of vaccines. Next one, all pathogens are parasitic, but all parasitic are not pathogen. The pathogen, the host pathogen interacts begins when pathogen in the form of inoculum comes in the contact and the inoculum causing infection when the inoculum causes infection when the host is susceptible and environmental conditions are favorable. When pathogen come in contact of the susceptible host, pathogenesis take place. The pathogenesis is the process of infection of disease development in a plant infected is the established of the pathogen within the host following develop. The pathogenesis is the most is the sum of a sequence of biological process in disease development. from initial contact between a pathogen and its host to the appearance of first symptom of the disease. A number of extra external and internal factors mm, such as the pre-penetration phase, penetration phase, post-penetration. These three phases are most important to the interaction of the host and pathogen. First one, pre-penetration phase. It is the host pathogen interaction before penetration of the pathogen into the host. The penetration is important only in the case of fungal inoculums and not in the case of viral and bacterium inoculum because penetration involves germination of propagule and growth of germ tube, which is the phenomena of fungal inoculums. Various type of spores like sporangia, conidia, odia, chlamydospores, and uridospores, ascospores, etc. and produced by the fungal pathogen. Some of them germinates immediately, whereas other germinates after a resting period. Various environmental factors like moisturizer, pH, temperature, light, carbon dioxide, and oxygen. These are the environmental factors. And coming to the chemical factors like host exudated residues and biological factors like microorganism present around the rhizosphere and sphere influence the germination of spores by antagonisms, competition, and microparasitisms. After encountering above factors, fungal propagule germinate by germs tube and penetrates directly into the host or it grows profusely over the host surface and are infection poisoned. The tip of the germ tube then form aspersoria, from which infected thread arise and enter into the host. Next one, penetration phase. It is the actual entry of the pathogenic into the host. The pathogen enters into the host either directly or indirectly. Indirectly, pathogen enters into the host either through natural openings present on the host surface, such as stomata, lenticel, hydatodes, or through the wounds. Directly, pathogen penetrates through uh, cuticularized epidermis. In this penetration, pathogen has to cross morphological, biological, and chemical barriers. The direct penetration is the first mechanical, then chemical. The germ tube of the fungal hyphae developed a presorium which exact a strong mechanical pressure on the wall. As a result, epidermis is stretched and become thin. The penetration takes place through this thin area. The penetration of cuticle is purely mechanical. The aprosorium extracts is a precision as at the thin area due to which cuticle breaks and pathogen comes in contact of epidermal wall of the host, which has pectin and cellulose. These are penetrated by enzymatic action of the hyphae, due to which the cell wall becomes soft and pathogen penetrates into the host tissue. At the same time, pathogen has also a neutralize many toxic substance created by host. Post penetration phase. In this phase, pathogens successfully establish themselves self into the host tissue, which is must for the appearance of symptoms. Various internal factors try to inhibit the growth of pathogen inside the host. The pathogen establish themselves after overcoming the internal barriers, which may be morphological and biochemical. The establishment of pathogen in the host involves a close biochemical relationship between the host and pathogen, between the two, that is the host and pathogen. Various enzymes, toxins, and growth regulators are created by pathogen mm, inside the host. It brings various morphological and anatomical and physiological disturbances in the host tissue. The physiological process such as membrane permeability, photosynthesis, respiration, and nitrogen metabolism are affected. The above distributions are expressed on the host surface in the form of the symptoms of the disease. On the basis of the infection, that symptoms may be local in which pathogen infect the host organ or part of organ, or maybe systematic in which entire plant is affected. The factors that establish infection is quantitative, larger the dose of infective pathogen, greater are the changes of infection. And post-postulated pathogenic test, Roth coach was German physician who confirmed the germ theory of disease and formulated four postulated known as postulated. These postulated are four criteria designed to establish relation between the microbe and diseases. The postulated, yeah, the postulated that was is the has been critical, important in establishing the criteria whereby the scientific community agrees that the microorganism causing a disease. In the post of first laid down 1882, a three-step series which he said must be fulfilled before the organism can be confirmed as the real cause of the disease. Later on, a fourth step was appended, first series of the conditions by E.F. Smith in 1905. These four conditions are four postulations known, four postulates universally accepted to establish a relation between microorganism and disease. These postulated are as follows. The organism must be found associated with the disease in all the diseased host examined. Second one, the organism must be successfully isolated and grown in pure culture or nutrient media. 8. Growth characteristic be recorded. The organism should be isolated from pure culture and must be isolated on the healthy host on which the disease appearance, and it must cause the same disease on the inoculated healthy host. The organism must be isolated from experimentally diseased host during the step 3 and grown in pure culture again. The organism grown in a pure culture must store exactly same characterized of its growth, which is shown during the step 2. If all above steps postulated are proved to be true, the isolated organism is identified. The next classification of next one is the host and plant affected. The culture and plant affected. First one is the host, casual organisms. Casual organisms. Next one is a part of the affected. Plant affected. And second one is called the part of the plant affected. Uh, and occurrence of disease. Fifth one is the natural nature of dispersal medium. These are the main affected. The first one is the classification of a plant disease. Classification of the plant disease on the basis of the host plant affected. Mm, This is the disease of the two types. One is the Systematic disease. Systematic disease in this case, Uh, in this case, whole plant is affected by the pathogen and the symptoms of the disease appearance in the whole plant. Localized disease when certain organs such as a root, stem, leaf, etc. are part of organ of the host plants affected, then it is called a localized disease. In this disease, the symptoms produced by pathogen on host surface are localized. The classification of plant disease on the basis of the causal organism. On the basis of the nature of causal organism, disease is the two type. One is the non-parasitic disease, second one is the parasitic. This disease is not caused by living organism. It is due to environmental conditions like low temperature, very high temperature, very low temperature, unfavorable soil mixture, moisture, soil pH and pH, atmospheric pH, presence of the toxic gaseous in the atmosphere, excess and deficiency of minerals in the soil also cause non-parasitic disease. As there is no involvement of living organism, it cannot be transmitted from one diseased plant to another healthy plant. Thus, the disease is non-infectious. The tip root rot or necrosis of mango is an example of non-parasitic disease. It is caused by deficiency of boron. The parasitic disease is due to the parasitic organism like algae, fungi, bacteria, and nematodes. The disease is infectious and transmitted from diseased plant to healthy plant in the same field. From one field to another field and from one place to another through various agencies. White rose disease in crucifer caused by the albugo candida is an example of parasitic disease. The third one, classification of plant disease on the basis of part of the plant affected. On the basis of the part of the host plant affected, diseases may be classified as the foliage disease, stem disease, root disease, vascular disease, and fruit diseases. That one. The first one is the foliage disease. When leaf of the host plant is affected by pathogen, that is called the foliage disease. Coming to the second one, when stem of the host plant is affected by pathogen, stem disease. The third one, root disease. When root of the host plant is affected by pathogen, root disease. Vascular disease, when vascular region of the host plant is affected by pathogen. Next one is the fruit disease. When fruits of the host plants are affected by pathogen is called the fruit disease. And classification of the plant disease on the basis of occurrence of the disease, the study of plant disease in relation to their occurrence, interaction of population of plants, pathogens and environment is known as epidemiology. There may be infectious disease and contagious disease, a disease which spreads slowly and is incited by transmissible pathogen is known as infection disease. And contagious disease. On the basis occurrence, disease are classified as a endemic disease, a disease which constantly occur from year to year in moderate to severe from an endemic disease. For example, the wart. The wart disease of the potato potato caused by the Syncytrium endobioticum is endemic to Darjeeling. In plant pathology, this term is applied to simple interest disease which are either indigenous or ascent introduction. Epidermis or epiphytic disease. This disease occur widely but periodically in an destructive form. The pathogen may be present in the locality, but the environment favorable for development of disease occurs only periodically. Thus, the disease is very responsive to variation of environmental condition, which actually control its incidence, cereal rust, powdery mildews, example for the epidermal disease. Next is the sporadic disease. The sporadic disease is The sporadic disease is rather an epiphytic disease, except that it occurs only here and there at irregular intervals. In an sporadic disease, pathogen affects only a few plants in a large population of host plants, and other remain unaffected. Angular leaf spot of an best example for the sporadic disease. Pandemic disease. This disease occurs all over the world and result in mass mortality. Late blight of potato is the best example for the pandemic disease. The classification of plant disease on the basis of the dispersal medium of the pathogen. On the basis of the dispersal medium of the pathogen, disease are classified into the soil-borne disease, seed-borne disease, air and wind-borne disease. The soil-borne disease, the uh, pathogen serves in the soil and disease spread through soil. Wilt wilt arhar in an example of the soil-borne disease, Fusarium species. The pathogen of wilt diseases survives in the soil. Seed-borne, the pathogen of disease survive in the seed. Therefore, disease spread through infected seed. Low smart of wheat is an example of this disease as the pathogen. Eustilagigo tetracy spread through the seed. Air and windborne disease. The pathogen disease survive in air, therefore spread of disease take place through air. For example, the iridospores of Vaccinia comes through air, infect the wheat crop and causing black rust of diseases. Next one, psychopathological terminology. The term used in the study of plant pathology carry defined meaning. Some such terms as follows. The different terms are follows the host one. Now, on which pathogen survive, that is called the host. And pathogen, a living entry which causing diseases. That is the pathogen. And the next one is the inoculum. The pathogen survive from outgrowing season to the other in the form of spores, um, Celerotia, dormant mycelium, or in various in various other in various other forms. This constitute inoculation. For example, inoculum in bacteria is the entire cell. In virus, the entire practical. In fungi, vegetative hyphae and spores. Thus, the part of pathogen which uh, which entire the host cells of the causing disease is known as the inoculum. Inoculum is two types. Primary inoculum, the form of a pathogen through which initiation of disease takes place is known as primary inoculum. For example, in white rust of cruciferous disease, the primary inoculum is oospores present in the soil. Secondary inoculum. After infection on the host surface, pathogen produced fresh crop or of spores which causes secondary spread of disease. These diseases are called secondary inoculum. For example, in the disease cycle of white rust of cruciferea, that is called albugo candida, that is the pathogen. Produced conidia mustard plant, that is the host. These conidia are secondary inocula as they cause secondary spread of disease. The disease, any deviation from the healthy conditions which interference with the normal structure and performance of vital functions of a plant is called disease. The disease plant can thus be spotted by changes in their structure or physiological process. The symptoms, external sign on the plant which indicate disease is known as the symptoms. Simple interest disease. Simple interest or monocyclic disease increased mathematically similar to simple interest on money. This disease develops from a common source of inoculum. That is the capital which is constant is known as monocyclic disease as there is one generation infection in a season. Compound interest disease. Compound interest disease is also known as polycyclic disease. This is mathematically analog to compound interest on money and characterized by multiple generation of spore being produced in a year. Etiology. It is study of living and non-living environmental causing of the plant disease. Pathogenesis, it is the study of the mechanism, disease development by pathogenesis and is concerned with the process of infection and colonized of host by the pathogen. Epidemiology is the study of the spreading of the pathogen within crop areas during the growing season. Epidermology has a wide meaning and he is concerned with the interaction of crop pathogen environment control. It is the state of the development. Sustainable method of controlling of the disease, reducing the loss caused by them. Antagonisms. The inhibition of growth of one organism by the other, which may be due to antibiotics for competition for food, etc. Bio certain organisms obtained their nutrition from living organism only complete their life cycle within the living tissues are called biotrophs. Example, rust, must, powdery mildew causing the disease by the fungi. Next one, elicitus, chemical signals generated by an organism to bring about metabolic shift in an another organism. The term elicitus is applied in phytology production. Fungitoxic antifungal substance which are harmful for the growth of fungi are known as fungitoxic. So fungicides. Fungistatic. Fungistats are compound which prevent growth of fungus as long as fungus and fungistat are in constant. Hypersensitivity, the extreme degree of sensitive of the host plant to the pathogen, resulting in the death of the host cell in the vicinity of the invading pathogen, thus blocking the advantage of pathogen. Inoculum, the part of the pathogen which enters the host cell and is capable of causing disease. Inoculum bacteria is the entire cell. In virus, entire particle, and vegetative hyphae spores in fungi. Infections, the establishment and nutritional relationship between the host and pathogen is known as infection. When the pathogen spread throughout the plant in varying degrees and is associated with all phases of its life cycle is known as synthetic systematic infection. Evasion and colonized. After entering or infected the host cell, the pathogen slowly begin to attach the neighboring host cell and beginning to establish itself within the host tissue by rapid division. This is known as colonization. Incubation, the interval of time between infection and appearance of disease symptom is known as incubation. Incubation period varies with host pathogen and environment condition. Immune, a plant which can resist or overcome disease infection said to be immune. Latent infection, the dormant state in which a host is infected with a pathogen but does not show any symptoms. Lesion, it is a localized area of discolored disease tissue. Mosaic, it is a viral disease leaves characterized by intermediate patches of normal green, light green or yellow color. Necrotic. The term necrotic refer to the tissues which are dead and discolored. Mm. Non-infection disease. Disease caused by the environmental factor and not a pathogen. This disease cannot be transmitted. Parasitic. Organisms that obtained their nutritional requirement from living plant hosts are called parasites. Parasite may be obligate parasite if restricted living tissues only, facultative if growing in living or dead tissues. Pathogen and pathogenicity. Any organisms, part or entire, which can infect another organism, that is host, distributing the host metabolism is known as the pathogen, and the ability of the pathogen to cause disease known as pathogenicity. Resistant, mm, the extent to which a plant prevents the entire invasion and colonization of a pathogen is a measure of the plant resistance. A plant is said to be immune when it shows high resistance and susceptible when it shows low resistance to the pathogen. Reorganization system, the earliest time when molecules of the host cell recognize the molecules of the pathogen and accept it for establishment. Saprophytes organism which derive their nutrient from dead organic matter. Suspect a plant that can be attacked by a given pathogen, that is the host plant. And suspectability, the inability of a plant to resist the effect of a pathogen. Symptomless carrier, plant which produce no obvious symptoms in spite of being infected with a pathogen. Systematic pathogen, chemical which spread internally throughout the plant body. Syndrome, the set of a various symptoms characterizing any disease, is termed as the syndrome. For instant of a disease may be characterized by necrosis of tissues, building and heterotrophy. All these symptoms collectively known as the syndrome. Phylophere. This term refers to the total above ground surface of plant when viewed as habitat from microorganisms. Rhizosphere, the soil surrounding the plant roots when viewed as a habitat for microorganisms. Antagonism refers to the action of any organism that suppresses or interference with the normal growth activity, plant pathogens such bacteria, fungi. Competition and interaction between organisms or species in which both the organisms or species are harmed. Competition may be intraspecific among the member same species, interspecific between the individual different species. The summary, this is the total symptoms. Tomorrow we will continue on the class, the topic name, symptoms and plant disease, 34. నేను పెడతాను. Okay. Tomorrow we will continue, ma. Yeah. Sir, 1 second. టేబుల్ మీద పెట్టండి, నేను సంతకం పెట్టి పంపిస్తాను. చేస్తాను నేను సార్, పెట్టేసేయండి నేను ఇస్తాను. If any doubts, okay. Mm. Yeah. Mm. Here, main important of the classification plant diseases and necessary for pathogenic disease and symptoms of fungal diseases. Second one, bacterial disease, viral, bacteria, fungi, viral diseases. Term. The main important of these are diseases under control to the pathogenic. And tomorrow, the 34 pages to start. Yes. Uh, Heinrich annotated Bury is considered father of plant pathology as well as founder of modern mycology. Edwin John Butler is considered father of plant pathology in India. Dr. K.C. Mehta and R. Prasad worked at epidemiology of central rust in India. T.S. Sadashivan established the School of Mycology and Plant Pathology at Madras University laid a foundation of research of soil-borne pathogen in India. The professor Millardet Bodex discovered Bodex mixture, which is a mixture of the copper sulfate and limestone. This mixture was used to control the downy mildew disease and grapevine disease of the grapevine. The discovery of the tobacco mosaic virus, that is called TMV, by Mayer in 1886 was the birth of the study of virus as pathogenic agents. In the 20th century, major trend in the areas like physiological plant pathology, genetics of host and pathogen, fungicides, environment in relation to plant disease, disease resistant in plants, biochemist and physiological of disease state, tissue culture in plant pathology, as well as ecological study of phytopathogens. All pathogens are parasite, but all parasites are not pathogen. The parasite which cause disease on host are called pathogen. The potential capacity of infection of any pathogen is called pathogenicity. The process of pathogenesis as their phases, pre-penetration phase, penetration phase, and post-penetration phases. Symptoms of disease appearance on the host surface after pathogenesis. The four criteria of host postulated necessary in established relation between the microbe and diseases. Here, the pathogenic test. The disease may be classified in as symptoms and localized, epidemic and epiphytoic, saprodic, pandemic, soil-borne, seed-borne, and air and wind-borne. The host is the plant on which pathogen survive. The pathogen is a living organism which cause disease. Symptoms are external sign on the host plant which indicate the diseases. These are the total summary of the diseases. Tomorrow we will continue on the diseased plant that is called symptoms of the plant diseases. 34.